All right, so basically to bring you guys back to, I like the small level, I like the microscopic level. Yeah, I kind of look at the human data, but I really like the cells. So and to kind of get us all on the same page, so this is a picture of the macrophages eating, phagocytizing the fibers. So there's little fibers in there. This is called the frustrated phagocytosis, where they eat these fibers and then can't really deal with them. And so all of these cellular and immunological responses occur. And so we have here, so asbestos is inhaled, gets into the body, macrophages attempt to remove the fibers. That doesn't really work. They are damaged and they start to die, and they start to release our cytokines and our inflammation markers, our reactive oxygen species, and we get these cellular responses that we think are directly related to fibrosis. And so we get the cells starting to put down collagen. We get the phenotype changes of our cells, and this leads to our fibrosis. But then we think these cytokines and inflammation also drive our immune responses. Then more macrophages and neutrophils come in, the B cells and the T cells get activated, we get these autoantibodies, and we get this autoimmune disease. So our ongoing question is, do the immune responses drive fibrosis? Do the cellular responses help drive autoimmune disease? So is there an interaction between these pathways? And we've already presented some data that suggests, yes, there's probably some interaction. But what exactly are these interactions? Okay. So Jean just actually went over this slide, so we're probably going to skip it. Basically, the mesothelial cell autoantibodies. Oh wait, go back one slide. Oh, you really can't see that. Let me hit the lights. You still really can't see that. Okay. All right, well, this was a really cool slide that you guys can come look at my computer if you want to see. You can kind of see this one right here. The blue on there, these are DAPI, this is a stain. These are nuclei of mesothelial cells. This green were our MESCA antibodies. So they're actually binding all along the surface of the mesothelial cells. So this whole slide shows a bunch of mesothelial cell autoantibody binding. It looks really awesome. And then when we cleared it, all that binding went away. So we took out the IgG, that all went away. So our mesothelial cell autoantibodies are an IgG antibody. And we had 18% of our Libby subjects tested positive for these. Yeah, it's still kind of iffy. And this was 4.88 times more likely to have a pleural abnormality if they had the MESCA. Okay? So again, this correlates with Jean and Brad and Tracy and Corinne's current work, as we think there is a correlation. We think that these antibodies, these mesothelial cell antibodies, are contributing to disease. We don't think they're just like a marker of disease. We think they are pathogenic. Okay? Okay. Because they make collagen in vitro, I already talked about this, so go on, and in vivo. So again, this is the same thing Jean just presented, but we took the mice, exposed them to asbestos, took the sera, put them in the other mice that had never seen asbestos fibers. That's really the crux of it. These mice had never seen asbestos. They've never been exposed just to the antibodies, just to the sera, and they made this collagen. So we used a multiphoton microscope to actually show this in the peritoneal cavity. So this is our saline exposed mice, so these are saline up here, versus our Libby amphibole exposed mice. This is the collagen. We can actually see, we can visualize that there's more collagen with our mesothelial cell autoantibodies in the mice. Okay, so this is a little bit different data, but this is the saline. We did 1 dose of our sera versus 2 doses. And you can see that we got statistically more collagen deposition when we added that second dose. So kind of repeating what we think is a MESCA exposure increased that collagen. But then really interestingly, when we took all the IgG, all the antibodies out of the sera and exposed the mice, we lost that collagen deposition. So we don't think it's just something like, oh, there's some inflammatory signal in the sera that's driving this. We think it is the MESCAs, it is the antibodies that's driving the collagen deposition directly. Okay? All right, so with that, kind of following up, and this kind of, I guess, goes to Ari's question is, well, what's next? What are you going to do next? So part of this is we want to identify more of our MESCA targets. We do think plasminogen is a target, and probably contributing to this overall mechanism, but we don't think it's the only target. So Zach is working right now of looking for these cell targets, again, with the idea that we might be able to find a pharmaceutical inhibitor of the target or the antibody, maybe a monoclonal antibody that could block these MESCAs and keep them from binding the mesothelial cells and keep that collagen from being deposited. So maybe we can block that progression. It might be too late to block the production of the mesothelial cell antibodies, but maybe we can block the progression, those rapid progressors. We could treat them potentially. Okay? And then also to look at the cell signaling pathway. So to understand not only what's the target, but how is it affecting this change in the cell? And maybe even if we can't block it at the surface, maybe we can block or change the pathway along which it's working.
Can I ask you a quick question?
Yes.
When you talk about collagen deposition, is it just the production of collagen, or is the collagen
It's being produced and laid down. Yes.
In a way that would be consistent with the way it happens physiologically?
Yes, and it is collagen 1. I've looked at soluble collagen, and we don't really actually see a whole lot of difference in the soluble collagen. It seems like all the mesothelial cells kind of produce collagen and kind of send it out, but it is only when the MESCAs are there that it's taking that, remodeling it, and actually laying it down in a matrix. And so some of my other work looks at some of the collagenases and some different enzymes that are remodeling enzymes, and it looks like there's some contribution, but it wasn't like the light bulb, like that's the pathway. We do think it's altering some of the collagenases as well. So whether that's so whether that's turning them on or recruiting them to the cell surface so that they're interacting more with the collagen, still kind of a mystery. Okay, so another question that's come up is, are the MESCAs produced only from Libby-amphibole exposures? Okay? So we've seen the correlation with Libby-amphibole, but what about chrysotile? We talked a little about chrysotile. So do they make MESCAs? What about the other amphiboles? We know they make the ANAs, right? But what about the MESCAs? And then we've also started looking at this non-asbestos exposed group. So back to Raj's question is, could this be a marker for asbestos exposure? Well, if so, then we have to make sure they're not being produced by non-exposed people, right? Okay, so the first thing we did is we took sera from Libby, amphibole exposed, and chrysotile exposed. These are, again, the Mount Sinai, the steam fitters. And we looked just for MESCAs. And we saw is a pretty similar prevalence between the two populations. So yes, MESCAs are being made following chrysotile exposure as well as Libby-amphibole. So it's not specific just to Libby, okay? But one of the really interesting things is then we looked at the pleural positive, so that correlation between having the MESCA and having a pleural change. And in Libby, we saw a strong correlation between those two, but in the chrysotile exposed, we didn't see a correlation. And when we were looking at pleural, we were just looking at any pleural changes. Okay, so not just like the thickening, but pleural plaques as well. So any sort of pleural disease. And we really did not see a correlation with the chrysotile. So they have the MESCAs, but they don't seem to be driving the disease like they do in Libby. And so the question here then became, well, is there something different about the mechanism? So are the antibodies there, are they doing different things? Okay. Oh, dang, I don't have a graph for that. So we looked at, so that's okay, I thought we had it in there. We did look at the collagen deposition between the Libby MESCAs and the chrysotile MESCAs. And we saw some differences, not only in the isotypes, but also in the amount of collagen that they're putting down. So we do think that they're actually targeting different cell surface markers. So they're targeting something different is our hypothesis. That's currently what Zach is working on. Is going to look at the markers, the cell surface binding between the different MESCAs. And so, again, we'd like to do this with other amphiboles, but then we're also looking again at the non-asbestos exposed populations. So we just started this semester the screening project with Dr. Thomas Muslin at the University of Washington. He studies people with rheumatic diseases, so mainly RA. He sent us a bunch of serum samples. He sent us 24 serum samples from patients, and our question was basically, do these patients make MESCAs? So when I started reading more about the systemic autoimmune diseases, especially the diseases we see in Livy, so lupus and RA and scleroderma, it just kept popping up in the literature that there's a subset of these people, these patients that have pleural diseases that are unexplained. Like, sometimes they get these pleural diseases, we don't really know why, sometimes they resolve, sometimes they don't. And that's all the literature said. It was like, they get them, we don't know. And so I started thinking, well, maybe MESCAs are involved and we just never looked for them. And so we started to look for them. And I mean, Zach has the data over on his poster. I'm going to give just a little bit of away. What we found is that some of them have MESCAs. But we only were able to screen 24 samples, so we don't have any good statistical data about what that means. We know there's some differences in the amount of collagen that they put down. So it's hard because we don't know this target, so we can't block the target and then like, okay, so what happens after that? It's also really hard to kind of measure because we're just kind of measuring binding and then we're looking for activity by measuring collagen. And we're doing that by an ELISA. So it's not quantitative in terms of we can say, oh, look, this makes, this much more collagen. We're just looking, does it make that collagen matrix lay it down? And it looks like the chrysotile MESCAs don't stimulate that collagen as like a strong reaction. There's some collagen that seems less than LA, but they're binding. The other weird thing is they have a different isotype subset. So they all have IgG1, but the Livy MESCAs have a lot of IgG3, where the chrysotiles have less IgG3 and more IgG4. And you can follow if I say this wrong, because it's been a while since I've thought about this, but the IgG4 is more immunosuppressive, and IgG3 is more active and related to a lot of autoimmune diseases. Okay, so this is your reminder. This is where we started. We think we have some pretty good evidence, again, that the immune response is kind of driving fibrosis, that there's an interaction, but there's still a difference. Okay, so 2 clicks. And so what we think this is that if we trade just that general reaction and we put chrysotile in there, chrysotile, instead of just an immune response, we're really getting an acute immune response that resolves. So we don't see this chronic inflammatory, we don't see this autoimmune response. So we think there's some sort of a resolution, and this is why we're not getting then those later, more progressive diseases. The cellular responses might also be different. Yes, we're getting some fibrosis, but it's mostly the plaques. It's not that progressive disease that Brad was talking about. Where if we stick LA in there, our hypothesis we're working on right now is that we're getting an immune non-resolution. So we're getting this chronic or this sustained immune response that then is driving that autoimmune disease, which then contributes to fibrosis. And with that, we're getting different cellular responses that also contribute to that different pattern of fibrosis. So that's kind of where we are right now. This is our big overall hypothesis, which is great. I'm a cell biologist. I love looking at the cells and the mechanisms, but we need to do something with this, right? So we need a driving force. And that is, can we take this information, can we take our hypothesis and figure out a way to help patients with this? And so that's really the next focus, is can we alter this immune disease and this immune response to then reduce the chronic disease, okay? So we're working on 2 different hypotheses or 2 different mechanisms. One is to alter the immune cell profiles to then alter the disease course. And the second is to use a synthetic lignin that's found in flaxseed to reduce early inflammation and immune activation. Okay, so these are our 2 kind of big therapeutic projects we're working on right now. So I'll show you a little bit of preliminary data from this first one. So I'm looking mainly at B regulatory cells, and our hypothesis is that Livy and chrysotile differentially activate the immune system. Okay? So then we're getting different chronic disease. So we think LA is kind of overstimulates the immune response. We're kind of overactivating it. We're not getting that resolution, which is then going to lead to these autoimmune diseases. And so for anyone who does not have a PhD in immunology, so besides you two, everyone else, so B regulatory cells is kind of a newer identified subset of B cells, and they have different responses. So as the name suggests, they're regulatory, but what that means is something different depending on which other immune cell you're looking at. And no, you don't have to know what all of these different things mean. The important thing to notice here is that they produce a lot of cytokines that do a lot of different things. Sometimes they're inhibitory, so we can block things like T effector cells and our monocytes. Sometimes they're stimulatory, so they can actually increase T regulatory cells. So the overall idea is that you're regulating the immune response. So you get something like asbestos or an infection, you get an immune response. These regulatory cells are then activated to say, hey, let's bring that response back down, and we're all good. So we think that with Libby amphibole, we're not getting the proper B regulatory response. So the immune system gets stimulated, it goes up, and then there's nothing there to bring it back down. So it just keeps going off at this high level. And we have some in vivo data to support that. So if we look at, we're just focused here and here. So these are B suppressor cells or B regulatory cells. They're the same thing. And we look at lymphocytes and in lungs, and these are mice that were exposed intratracheally to asbestos fibers. 8 months later, when we have chrysotile exposure, we see statistically significant increases in B regulatory cells. With saline and Libby amphibole, we don't see those B regulatory cells elevated. So basically, we don't have a suppressive response with Libby amphibole. We do have a suppressive response with chrysotile. Okay. So right now, I am putting the finishing touches, hopefully, on an NIH R15 grant to submit to look at these activities, so the B regulatory cells. And do I have something else on here? Yes. The hypothesis, again, is that Libby amphibole blocks the production of B regulatory cells, so then we're getting too much immune response. And then we're going to try doing an adoptive transfer of active B regulatory cells. So basically, we're going to stimulate the B regulatory cells and transfer them into mice that have been exposed to Libby amphibole to see if those regulatory cells can lessen the disease progression. Okay, and so then this last project, again, is looking at kind of a therapeutic potential. We're looking at a synthetic flaxseed-derived diglucoside, SDG. Okay. So this was developed by Dr. Christophorou, Melpo, yeah, that's what we call it, at University of Pennsylvania. So this is a collaborative, she called Jean actually and said, hey, I got this flaxseed, we want to see if it blocks immune responses or the inflammatory responses in mice. It does in cells when we put asbestos on them. And she said, great, I'll look at this. And by the way, I might someday retire, and so you should talk to Kinta about following up on this. So I'm very grateful to have been brought in on this project. It's been a lot of fun. And so what they've seen in a cell culture is that this SDG has anti-inflammatory, antioxidant properties. It's a free radical scavenger, and it's a myeloperoxidase inhibitor. So this is an enzyme that is in neutrophils, largely in neutrophils. So these are some of our first responders of the immune system. And so what we did is very short-term in vivo experiment where we gave the drug 3 days prior to asbestos and then 3 days after, and then sacrificed the mice and looked at immune responses. So this is just a little bit of the data. What we saw is when we had control mice and LGM, so just the drug by itself, we didn't really get any immune responses. So we're looking at immune cell trafficking into the peritoneal cavity. So this is where the drug or the fibers were instilled intraperitoneally. And so we didn't really see much of a response except for when we had LA by itself. So the LA are drawing these immune cells, these innate immune cells into the peritoneal cavity. And then when we add the drug, we're blocking that response, that calling in. So, and we see this again in both of these cell phenotypes. So this again is all innate cells. We didn't look for our B cells and our T cells. We didn't see much in that, but again, it was only 3 days. So we don't actually think it was enough time to stimulate the adaptive immune response. And so Reagan, and call out each of my students. So Reagan is working right now on developing a 10-day trial so we can look at the adaptive immune response. And then we're going to follow that up hopefully with a 30-day trial, right? So she's got the poster. Okay, so we just put in an R01, NIH R01 application to look at this in more depth. This went in February, so hopefully getting scores next month, so cross your fingers for us. So we're looking at the beneficial effects of this drug. Tracy, you wanna? And our hypothesis is that early treatment, that B should not be there, with LGM will reduce early immune activation and inflammation, which will then reduce long-term disease outcomes. So my part is looking at the autoimmune and the fibrosis. We also have another collaborator looking at mesothelioma. So we're hoping this not only help with the immune responses, the autoimmune disease, but also with the cancer responses following asbestos exposure. Yeah. Okay. So so in summary, we're looking overall at reducing early inflammatory responses and early immune responses as a way to control those more chronic diseases. So we're trying to identify and block the MESCA targets. We're looking at altering our B regulatory cell populations, and then we're looking at using LGM as a chemopreventative. So if you know somebody's been exposed to asbestos, perhaps they can start taking this drug before the disease develops to block that early inflammation. All right. Well, thank you.